Welcome back to the next video in Introduction to Neuroscience. Now in this class, we don't have a track record of shying away from touchy subjects, and we are not about to start now. This video is going to be all about somatosensation, the sensing of your body. Now somatosensation can be broken down roughly into 3 different classes of senses, and they have slightly different mechanisms and different pathways that are involved in your nervous system. So we're going to be talking about each of these in turn. We'll start out with your sense of touch, move on to your sense of proprioception, how do you know where your body is in space, and then we're also going to be talking about temperature, pain, and itch as a slightly separate category of senses in somatosensation. Now, somatosensation is one of the more complex and least well understood of the senses. And relative to our understanding of things like vision and auditory senses, there's just relatively little that we know about somatosensation, and there's a couple of reasons why this is true. The first reason is, as I said earlier, somatosensation is about sensing of the body. And so rather than having localized sensors that are in one place, like all of your photoreceptors are in your retina, all of your hearing cells are inside your cochlea, sensors involved in the somatic senses are distributed all over your body. And not only are they distributed all over your skin, all over your body, they're also inside your muscles. And this also includes sensing of the internal organs. Something if you hear the term interoception, that's something that's about the somatosensation of the internal organs. And so this is a difficult topic in part because the primary sensors involved in somatosensation are distributed in a lot of different places. And not only are they found in all of these different places on your body, they also involve a lot of different types of sensory neurons. Again, in contrast to photoreception, we know that in humans at least there's rods and cones, and there's 3 different types of cones, but even among those 4 different types of photoreceptors you express in your retina, they're all relatively similar in the grand scheme of things. They all involve rhodopsin, and they all involved this cis to trans retinal phototransduction pathway. In contrast, as you'll find out in the remainder of this lecture, there's a ton of different sensory neurons and different sensory transduction proteins that are involved in somatosensation, and so that makes it really challenging to study as well. The other reason, as you'll see, is we talked a lot about in the beginning of the set of lectures on sensing and action, about how it's actually not sensing and action, but a tightly integrated sensory motor loop. Now, this is also true in vision because, after all, there's active vision. You can move your body around actively to seek more visual input, and also seeing something allows you to guide your movements in the future. But this type of tight coupling between sensation of movement is nowhere more obvious than in the somatosensory senses. And it's because the movement of all the different types of different neurons are in tight feedback loops with your motor system. That's one of the most rapid senses, and it feeds back onto the motor system with the shortest latency. And it's also something that we don't really consciously think about very much. Like if you're hiking and you step over a rock and you trip over something really quickly and you correct, it's something that you do without really thinking about it. And it happens in a very, very rapid time scale. And so that makes it so that it's really hard to study somatosensation when the animal is immobile. Now, as you know from the lecture on electrical and optical methods that we use in modern neuroscience, the vast majority of those methods involve the animal being relatively immobile. If not completely immobile, then at least somewhat restricted to a small arena. And so having a sense that is so tightly coupled with movement makes studying it more challenging just from a technological perspective. And it also makes it so that it is recordings done when movement is happening is prone to all kinds of different artifacts. So even electrical artifacts, optical artifacts associated with the body moving around, all of these are kind of technical challenges associated with studying somatosensation that makes it so that we can do it, but it really involves a whole brand new set of different tools, both technological tools as well as computational tools to really deal with the type of data that we're getting out of studying of the somatosensory system. But we do know more than just a little bit. So I talked about the 3 different types of somatosensory senses: touch, proprioception, temperature, pain, and itch. Now, a majority of these are associated with a mechanical stimulus. So this has to do with forces, stretch and strain, for example. They come with different kinds of peripheral endings, and are different proteins that are involved in each of those different types of cells. Some of those cells have very interesting geometric specializations that make them suitable for detecting that particular type of mechanical force force. There's a lot of geometry and morphology involved in the study of somatosensation. In the case of temperature, there's also there's some sensation of heat and cold and chemical senses, and so that's a whole new different class of molecules that are involved in that detection, and they're often found as free nerve endings and on your skin as well as in internal organs. And so the study of somatosensation is kind of lumping all of these things together, and so we'll see throughout this next couple of lectures what they have in common, as well as what are some details we know that make them different and distinct from each other. Okay, so I told you that somatosensation is a sensing of the body, and so how is the nervous system of the body organized? We mentioned this a little bit in the lecture on the organization of the nervous system, but I'll bring it up here one more time to tell you a little bit more about the details. So your brain and your cerebrum is up here, right? And the whole thing kind of projects down through your spinal cord, and what happens is that you have these vertebrae, right, that are in your spine, and there are these nerves that come out in between every single one of these vertebrae, and they go and innervate the rest of your body, going outwards. Okay? Now they're organized in the following way. There's some words that I'm going to tell you about with which we refer to these vertebrae. So there's the cervical vertebrae, thoracic, having to do with the middle part of your body, lumbar, kind of the back, and then sacral, kind of towards where you would have a tail if humans had tails, but this is where a dog or a horse would have a tail. That's kind of the sacral version of segments of the spine. There are canonical versions of these, and they all have numbers. They start with 1, and they go towards whatever they are, right? In humans, these are the numbers that apply in the human case, but there's, of course, a substantial variability depending on the animal you're talking about. One funny fact that I thought was really interesting is that the vast majority of mammals have 7 cervical vertebrae. So those are the vertebrae that go from the first one that's right underneath your brainstem to the last one that's right above your shoulders. Okay, so you and I each have 7 cervical vertebrae, and all mammals have 7 vertical vertebrae, including this gigantic giraffe here. So if you've ever seen a giraffe in a zoo or like a skeleton of a giraffe in a museum, you can actually notice that they have 7 gigantic cervical vertebrae. They're each like this big. It's really crazy to look at. In contrast, in avian species, they also have really long, exaggerated necks. But among the avian species, the number of cervical vertebrae can vary anywhere from 11 to 25. And so there's a lot more variability in terms of the organization of the cervical vertebrae in avian species. There's a couple of other really funky spinal specializations of birds that I love nerding out about. Maybe I'll tell you about that on a separate occasion. Okay. So that's the organization of the different segments of your spine. Now, each segment has the following organization, and it kind of repeats, right? So it's not exactly the same, but each canonical spinal segment has the following structure. It is bilaterally symmetric, as you may expect, and so there's a midline, and there's what's called a central canal down the middle through which cerebrospinal fluid flows. And it has a left half and it has a right half. There's a bit of white matter through which all of the cables run up and down towards your brain and away from the brain. And it has what's called a dorsal root ganglion, the DRG. And the dorsal root ganglion is where the cell bodies of all of the sensory cells that innervate the rest of your body live. So it's one of these funky cells where the cell body actually lives in the spine, in the dorsal root ganglion of the spine. One end of it goes to the patch of skin or muscle that it innervates. Okay? And the other end of it projects all the way up towards the bottom of your brainstem. So the dorsal root ganglion is where the cell body, the sensory cells live. As you'll find out in one of the later lectures, the ventral root is where all of the motor cells live. But by the time you get to these nerves that are leaving the spinal cord, the sensory and motor nerves are intermingled. They're kind of in the same bundle, so you can no longer distinguish them that way by the time you have a bundle of nerves leaving each of the spinal segments. A bit more organization here, that is a consequence of this organization of the human spine. You can kind of map the patch of skin to which each of the spinal segments go to and draw a map of the following way. It's called a dermatome because it is a map of the patch of skin that go to the same go to the same spinal nerve. So for example, here we were talking about the cervical nerves before, so you can see C1 is, well, actually no C1, whatever. It starts with C2, C2, C3, C4, C5, C6, and 7, and they are all responsible for innervating strips of your skin, starting from the neck and the back of the head, and then all the way going back down to most of your arms. The majority of your arm is innervated by the cervical spinal nerves, and the only one that is innervated by T1, the first thoracic segment, is this strip of skin right here. And you can kind of see what it looks like on the back of your, in your kind of your upper back area as well. Now, these dermatomes are interesting, not only because they're true, and this is interesting, it's actually how it works. The other consequence of the dermatomes is if you think about a viral infection like chickenpox. Now, chickenpox is a type of herpes virus that makes you really itchy. I caught it when I was a kid, and what happens after you recover from the major primary symptoms of chickenpox is that this herpes type virus, the herpes zoster virus, actually takes up residence in all your peripheral nerves, like in the nerves that go from that patch of skin to the spine. And it kind of just hangs out there and it lives there for the rest of your life until and if you are usually as an older adult or under a tremendous amount of stress. Sometimes people get these with an immune compromise. They reemerge as shingles. Surprise! The distribution of shingles, because of the fact that the viruses live in your peripheral nerves, will often highlight one of these dermatomes. Like you'll have somebody who has shingles not everywhere on their back, but one strip that corresponds to like T3 or T4 or whatever it is. It'll have that geometric organization that highlights exactly, delineates the dermatome on that individual, because it's the herpes viruses that are reactivated in that particular peripheral nerve that have flared up and caused shingles. So it's kind of a interesting but not exactly charismatic way of visualizing these dermatomes. The other question you might be asking yourself is, this is kind of weird, right? Like, why is it like this? Why is this one strip of T1 that innervates like your forearm area? And why is it that the front of your leg is innervated by all of these L and S segments? The back of your leg is entirely different. This makes no sense, right? And it really doesn't make sense. And it only makes sense if you think about it in terms of animals that are not bipedal like we are. And so if you do one of these moves where you try to touch your toe, you will see that your dermatomes then line up almost precisely in terms of the anterior to posterior axis of a quadruped animal. And so just like the orientation of the brain in terms of what is anterior and what a coronal section is and what is front and back in the brain versus the rest of the body and the spine, the organization of the dermatome can be explained by the fact that they were established when we were not a bipedal species. And so it totally makes sense in that context. You're going to think about where they are, why those strips are where they are. That's the reason why they are where they are. Okay. So we talked about the dorsal root ganglion cells, and these are the sensory cells that innervate your skin as well as your muscles. So there's a couple of different things that I wanted to highlight here about a little more finer detail organization of the dorsal root ganglion. I'm highlighting here that there's different classes of somatosensory neurons whose cell bodies live in the dorsal root ganglion, and one end is the primary sensory ending, and they go to different parts of your skin and comprise a large class of different touch sensors. We'll get into exactly what they are later. There's also a preview of what's coming up in the next section of the video about proprioceptors. These are proprioceptor spindles, muscle spindles that are actually embedded in the middle of your muscle, and they are responsible for measuring the length and rate of change of your muscles. And so every time your muscle gets longer or shorter, these spindles will fire in proportion to the length and the rates of change. And so all of these mechanosensory neurons are going to be projecting to the dorsal root ganglion cell. But they're not all the same. And one of the ways in which they're different is the extent to which they are myelinated. Now, if you remember back to the lecture about the cable equation and conduction by myelinated and unmyelinated axons, I previewed a little bit there that not all of your axons are myelinated. In fact, the ones that are myelinated are usually the more important ones where you have to get that information really, really quickly. And then some other axons are in fact unmyelinated because you don't care about getting that information very quickly. It doesn't really matter, or it actually may even be advantageous for you to receive that information more slowly. Well, this is the best example of that. So among all of the nerves that come from your dorsal root ganglion cell is a variety of different axons, some of which are heavily myelinated, and then some of which, and they have different names, are not at all myelinated. If you look at the ones that are heavily myelinated, they include these proprioceptor neurons. And the proprioceptor neurons have to project very, very quickly as possible to your spine, because this is your sense of update of where your body is. This is the only way, if I close my eyes, that I know where my arms are. And so these proprioceptor neurons, the muscle spindles, are one of the more heavily myelinated parts of your nervous system, because it is so crucial that they get the information to where they're going very quickly. Okay, so we talked a lot about these types of neurons, and so now we're going to start characterizing them. In the same way that we characterized visual cells in terms of the receptive fields, we're going to expand on that topic here and use the same terminology and the same approach to describe somatosensory neurons. Like visual neurons, somatosensory neurons have a spatial extent of their receptive field. Now, the receptive field in this context literally means what part of the skin do you have to poke on, do you have to touch, to get that somatosensory touch receptor to fire. So it is a receptive field in this actually a physical map that has a picture of that part of your body and with a dot on it, and it says, okay, this is my receptive field of this tactile neuron here, and it means if I poke at it, that's when it's going to fire. Okay, very, very intuitive. It's very like literal, a spatial receptive field on the space of your hand. Just like we had spatial receptive fields for visual neurons, which was usually a location in visual space, like flash of light here, I'm going to activate one neuron, flash of light here, I'm going to activate a different neuron. Similar here, if I poke here, I'm going to activate this one touch receptor neuron, and if I poke somewhere else, it's going to activate a different receptor neuron. Now, I've shown you pictures here of two different touch receptor neurons, and they're both touch receptors, but they're different types. They have names that are named after some of the individuals who are first responsible for describing them. So we have these Messner's corpuscles and Pacinian corpuscles. And you'll see immediately that these two different types have overlapping receptive fields. Okay, there's parts of your skin that are innervated by both of these types of tactile receptors. However, they are really different in terms of how large the receptive fields are. Messner's corpuscles have very small spatial localization. They're very sensitive. They're only firing when you poke that very precise piece of the skin, whereas Pacini's corpuscles have these gigantic receptive fields, which is like half of my hand, okay? So you can have these spatial receptive fields that describe how sensitive and which areas of your skin these different touch receptors are sensitive to. Now, different neurons talked about having different receptive field locations and sizes, so that's an illustration of those. They also can have differences in what is the threshold intensity that causes them to fire an action potential. So you can have, as a property of that tactile neuron, as a part of its receptive field, its sensitivity. So some of them might be really sensitive. You'll barely scratch it and it's going to fire. There might be other ones that are relatively insensitive. You have to really hit that piece of skin before it fires an action potential. The other way in which the receptive fields of these tactile neurons differ is the timescales of adaptation. Now, adaptation refers to the fact that when, in the absence of change in the stimulus, a lot of neurons will adapt and stop firing when nothing is changing. So it's reporting a change in the stimulus rather than an absolute value or intensity of the stimulus. And that is also very much true in different types of tactile neurons. We'll have neurons with different timescales of adaptation. Some of them only fire right when you touch something, and others will continue firing as long as you're continuing to touch something. And so we have a variety of different receptive fields, and these are all the different ways in which they can be different. And these are features that we would characterize when we talk about them in terms of what's their favorite stimulus. How do you present a stimulus to this neuron to get it to fire for you? So these are a couple of different features. We talked mostly about spatial features, but these neurons also have temporal features, because after all, it's not just the location of the touch that matters, it's also the frequency. So here, again, we're talking about the same two different touch receptors, the Messner's corpuscle and the Pacinian corpuscle, and you'll see that on the horizontal axis here, we have the stimulus frequency. So this was interrogated when you basically get a little vibrating probe, right? And you find your little probe, you put it on the piece of skin, and you vibrate it at different frequencies. And that's logarithmic axis up there, so you can see that every time the tick mark goes up, it's actually going up by several fold orders instead of in a linear scale. 10 hertz means 10 cycles per second. That means I'm pushing down 10 times per second. And 100 hertz means 100 times per second, which is 10 times faster than 10 per second. Okay? Now, On the vertical axis, what we're plotting there is the threshold amount of skin indentation that caused that particular receptor to fire. So, also in logarithmic coordinates, right? This is in microns now. To unpack all of that information, the lower the threshold, the more sensitive it is, right? So the very bottom part of that sensitivity curve means that that particular tactile sensor is most sensitive at that frequency. And so the lowest threshold that activates one of these orange receptors is right here around 50 hertz. That means that that's its favorite frequency. If you hit it that frequency, it will have a very low threshold. It's very easy to get it to fire. Whereas for one of these gigantic Pacini corpuscles, it is super duper sensitive. You'll notice that the difference here is it's over tenfold. It's almost 100-fold difference in terms of sensitivity. So these Pacinian corpuscles have gigantic spatial receptive fields. They can't localize where the stimulus is in space, but they're significantly more sensitive, and they're sensitive at a higher frequency. So its peak frequency, its peak sensitivity frequency is at 300 hertz rather than this 50 hertz that we see for the other types of corpuscle. So those are all the different ways that we can describe receptive fields of these tactile neurons. So to summarize, different neurons have different receptive fields, and these receptive fields, you can characterize them in terms of a lot of different dimensions. And none of this is complete. No one dimension of characterization of the receptive field is complete. It's really a combination of these properties that makes it so that we can fully characterize what are the features that these particular tactile sensors are sensitive to. And we have to pay attention to both space and time. I talked already about Pacini corpuscles, and they're quite lovely. Also, if you partially they're lovely because their receptive fields are so large that they're actually macroscopic. And so if you look online, you can find videos of hand surgeons where you can dissect out a Pacinian corpuscle. They have this really cool shape where it's just like a myelinated axon with all the glia wraps. It's all these little wraps, and so it's kind of like a squishy gel pack, and that's how it achieves the exquisite sensitivity that it has in detecting different very high frequency vibrations. The Pacini corpuscle is also a really good example of what I'm going to call embodied computation. So in neuroscience, we talk a lot about neural properties, but of course, if you dig down into the properties, the mechanisms of transduction, you have to get down to the level of proteins, chemicals, as well as biophysics. In terms of the photoreceptors we talked about, spent a lot of time talking about the biochemistry of the retinal, also the way that it's stabilized by the rhodopsin molecules that surround it, because that is the basis in which it can detect photons. Similarly, if we're talking about receptors that are embedded in your body, we're really then talking about the biomechanical transduction channels that interact with the biomechanics of the cell, as well as the biomechanics of the tissue in which it's embedded. So all of these touch receptors are embedded in your skin, and that means the stiffness, the elasticity of your skin, as well as how much your muscles are bending, and how the relative orientations of your bone, those biomechanical details are very much important because they are the ones determining the stimulus in terms of forces that actually make that particular cell fire. So what I mean by embodied computation here is that the receptive field of that neuron is not solely a neural property or a protein property or a chemical property. It's very much a property of all of those things and the mechanics of the limb in which it happens to be embedded. And we need to know all of that in order to figure out what's going on. All right, I want to keep digging in, take a little bit more details about the mechanosensory touch neurons. There have a bunch of different types, and they're all embedded in slightly different places in your skin. So there's some of them that are in your epidermis, there's some of them that are in your dermis, and they have different names. And so we're just gonna, I'm less concerned about you learning the names of these exact receptors, but we are going to talk a little bit more about their properties and the different ways that their receptive fields are different. And so here we have 5 different mechanosensory touch neurons, and they have letters and numbers. Let's not worry about that for now. Just go with me for a little bit. There's 5 different types of touch receptors that are co-localized on your hand, on your skin. And here's how they respond to the same stimulus. So my stimulus is a probe that's going to go down, push on the skin, and then come back up. That's this curve down here. Okay, I'm going to push and then let go. Okay? So you can already see immediately that these 5 different types of receptors are going to respond very differently to the same stimulus. We'll have ones that are relatively slow adapting, where they're really responding by firing faster to the indentation. Okay? So they don't really adapt. They're actually persisting in firing faster when you push harder on them. On the other hand, we have, and among them, we have the small receptive fields and the large receptive fields, but that are both slowly adapting. Okay. We also have what are called rapidly adapting cells, also with the other two different types, small receptive fields and large receptive fields. And they're rapidly adapting because, yes, you can see they're only really firing action potentials as you're pushing down, and then they stop even if you're holding, and then they fire again when you let go, because they're really responding to that change rather than the intensity of the indentation. So we can call them like movement detectors and also vibration detectors. They can also come in different types in terms of how sensitive they are. These four are all considered low threshold mechanoreceptors. They're relatively sensitive. Their threshold for firing is pretty low, and you just have to barely touch them and they'll fire. And then we have ones that are relatively high threshold mechanoreceptors, and those have slightly different properties. And you can see that they fire in response in this very slowly adapting way, but they're different in that they have a higher mechanical threshold. And in terms of the circuitry of where they project to next and what information your body is getting from the sensor and what it does about it, that's sort of like a downstream circuit level question that we'll get to in a couple of lectures. Okay, so that was your sense of touch and the different receptors that are in your skin that are responsible for your tactile sense. Next, we're going to move to the next somatic sense, which is proprioception. I've already talked a little bit about proprioception because you can't really separate the proprioceptive neurons from the touch receptor neurons in your nerves. So we already had a diagram where you can already see that there were these muscle spindles that are all wrapped up together with your touch receptors, and they all go to the dorsal root ganglion cells. But proprioception is a word that is it's a less common English word than touch. Everyone knows what touch is, but most people, until they take a class in neurobiology, don't really know what proprioception is. So I think it's worth taking a moment here to just demonstrate exactly what I mean by proprioception. Proprioception, broadly writ, is defined as your sense of how does your brain and your nervous system know about where your body is without looking at it. So the classic demo is the following. So what I want you to do is I take your two index fingers, and with your eyes open, you're going to make them touch each other. Okay? Hopefully this is something that is possible for you to do. Then the next thing I want you to do is try to do exactly the same thing, but with your eyes closed. So now I don't really see my fingers. Okay, kind of missed there, but I got close. Well, that was pretty good, right? My fingers are touching each other, or at least they're getting close to touching each other. So for me to accomplish that task without vision requires that my nervous system, my body, know about where this arm is and where this arm is in space. So that's my sense of proprioception. It's a set of mechanosensors that allows my nervous system and my central and peripheral nervous system to estimate the exact angle and kinematic trajectory of my arms, even when I'm not looking at it. Now, proprioception is a sense that is, I think, pretty underrated because we don't really think about it. We take proprioception for granted. And you only realize how bad it is when you don't have it because there's a couple of rare different neurological disorders where a couple of individuals have lost selectively only their proprioceptive neurons, and they basically can't do anything. I mean, they can't move their muscles are fine, the touch receptors are fine, their vision is fine, everything is fine, but to do even simple tasks like picking up an object, right, requires that you have this rapid feedback from your muscles and your joints about where that arm is. And if you don't have that, it's really hard to do it without staring at your arm. And so there are individuals who have, after a long trial and error, learned how to walk again. Because when you're walking, you don't really look at your legs, right? You just walk. You know, your legs know where they are and they do their thing. It's distributed. But if you don't know where they are because you've lost your sense of proprioception, then you can only walk by staring very intently and concentrating on where your legs are. So it is possible to walk without proprioception, but proprioception is absolutely vital for coordinated movements, for the types of very fluid, very natural movements that we all enjoy. Okay, so that's why proprioception is and why it's important. Let's dig a little bit more into the different types of proprioceptive neurons. Then in mammals, they come in three rough different types, and they're all important, and they do slightly different things. I've already told you about the muscle spindles. Now, muscle spindles are mechanosensory neurons that are embedded inside your muscles. And they are there so that when the muscle changes length, when you're contracting or you're extending your muscles, the muscle changes length. And as the length of that muscle changes, it either stretches or contracts, right, and changes length. And the muscle spindles are responsible for sensing the length of that muscle and its rate of change. Okay? So in other words, it's measuring both a position and a derivative of your muscles. And you have these muscles you have these skeletal muscles in which the muscle spindles are embedded. Not all of your muscles have spindles, but a large number of the skeletal muscles that are responsible for moving your joints do have spindles. You also, in your arm, had what are called Golgi tendon organs. They are located, as you may expect, in the tendons. They sense muscle forces and also its rates of change. So if you remember from physics, length is position, rate of change is dx dt, and then force is going to be proportional to acceleration. So it's going to be the one more derivative. It's going to be a second derivative of position is going to be proportional to the force. And so these Golgi tendon muscles are sensing muscle forces and its rate of change. In addition, you have what are called joint receptors that are actually embedded inside the joint. So where in your elbow joint, there's two bones that they fit together, and the joint receptors are actually inside that interface. And they're usually there to sense extreme joint positions, kind of when you hit the very end, right? You're fully extended, this joint can't go any farther. That's when the joint receptors are going to be activated. Now, these three classes of mechanosensory proprioceptor neurons have different biomechanical mechanisms, and they also connect to different pathways in terms of how they go to your spinal pathways, as well as where your body does with the rest of the information. How does it compute on this? How does it actually use that to determine how you're moving your arms? How are you walking? How are you locomoting? Stuff like that. This is the mammalian version, but interestingly, there's actually an analogous version of every single one of those receptors in the invertebrate system. Now, the names are all different, and the mechanical mechanisms actually are different as well, but there's analogous structures for the spindle, the tendon organ, as well as the joint receptors if you're looking instead at an insect leg rather than at a mammalian or vertebrate limb. Okay, so mechanosensation in mammals had a heyday, had a giant celebration a couple of years ago because Artem Patapoutian was awarded the Nobel Prize a couple of years ago for his role in the characterization and discovery of a class of proteins that turned out to be absolutely crucial for mechanotransduction in mammals. And the protein's name is piezo. There's piezo 1 and piezo 2. There's a couple of different types, and they're found all over the place. Absolutely vital for sensing of mechanotransduction in mammals. And so what you see there is a demonstration of that. When you have a mechanical stimulus, that is deflection of some kind, with piezo, the cell is able to fire a current. If you knock out piezo 2, then no more current. This is the necessary and sufficient for detecting mechanical deflections when you have a mammalian cell. Now, as you may suspect, like we talked about in basically every single lecture, this is a cation channel, and it is a mechanically gated non-selective cation channel. In the course of these lectures, we have seen voltage-gated cation channels, we have seen ligand-gated cation channels, we have seen photoactivated cation channels. Here is a mechanically gated non-selective cation channel. It is not the only non-selective cation channel that's mechanically gated. There's actually a variety of them that other species that are not mammals have, but this is the one that is vital in mammals, so that's the one that we have to do all of the things that we need to do that involve mechanical forces and stretch and strain and stuff like that. It is also involved not only in what you would normally think of in terms of movement, it's involved in a lot of different interoceptive systems. So here, for example, is a paper that demonstrated that piezo 2 is necessary for the proper function of the bladder. All right, so it's inside the bladder. These cells are in the bladder because when the bladder becomes full, you have that sensation of, oh man, I gotta go. So piezo is the protein that lives in the sensory cells of the bladder and gives you that sense of urgency and to completely changes your behavior, right? When you feel like your bladder is full and you have to go take care of the situation before you can do anything else. And so it's not just found in kind of these like gross anatomical movements, but also in interoception as well. So if you have like a sense of things moving around in your gut, maybe if you ate too much, maybe sometimes you feel things inside your inner organs. There's a variety of different nervous systems inside your internal organs. I'm not really mentioning them here, but they're actually super duper important. So piezo and a class of proteins that are related to piezo are all involved in those pathways as well. Okay, so that was proprioception, kind of your sense of where your body is and also what's happening inside your guts. Next, we're going to turn to the next set of somatic senses, and these are a set of sensors that sense temperature, pain, and itch. Now, the same year the Nobel Prize was awarded to Artem Patapoutian, it was also awarded to David Julius for his role in the discovery of trip channels. And not only, sorry, he didn't discover trip channels, but he discovered the role of trip channels in mammalian sense of pain and temperature. So trip channels are a very large class of proteins that are found everywhere. There are like lots of different types of these trip proteins and they're everywhere. And trip kind of stands for this kind of a, I don't know, not very explanatory name, the transient receptor potential channels. Now, trip channels are cool because they sense heat and coldness. So there's a couple of different types of them. I'm highlighting two of them here. There's trip V1, right, which senses heat, and then trip M8 is one of the ones that senses cold. So you kind of see the potentials here in terms of what happens with the currents when you apply cold. Trip M8 has an inward current. If you apply heat, the opposite happens. Interestingly, not only are they sensitive to cold and heat, these different temperatures, at the same time, they are activated by a variety of compounds that you are probably familiar with. So for example, menthol activates the cold trip channels, and capsaicin activates the hot trip channels. This is why when you are chewing on a piece of mint or has minty gum that you're chewing on, it actually feels cold. It kind of has this cold sensation in your mouth. It's literally because it's activating the trip M8 channels, the same channels that are activated by cold temperature. Similarly, when you are chewing on a chili pepper and it's really hot, right, and it's actually kind of painful, it's the same thing as being burned. Right? So the reason we call hot foods hot is because it's literally hot. It's actually activating the same V1 channels that also respond to hot temperature. I talked a little bit about this in a previous lecture when we were talking about gustatory senses. And so the taste of spiciness is something we mentioned. I told you the reason that it was not exactly gustatory sense is something we'll talk about in somatosensation. So here we are talking about it in the somatosensation lecture. Here is a variety of different trip family of channels. And you'll see that they actually tile the space in terms of the temperatures to which they are sensitive, all the way from very cold to very hot. And also, they are activated by a variety of naturally occurring compounds that we see in herbs and in foods, including, we've already talked about chili and we've already talked about mint. So mint activates trip M8, which is a cold receptor, and then chili capsaicin in chili activates trip V1. It also activates trip V2, which is even hotter. Its receptive field in temperature is even hotter than trip V1. Now, if you think back on that demo I did with the jelly bean in the chemical senses lecture, I told you that there's some jelly beans where this demo does not work. And the reason that it doesn't work is because the flavor of that jelly bean is not activating an olfactory receptor. And it's not activating an olfactory receptor, and I told you cinnamon was one of the ones where it doesn't work. And the reason cinnamon doesn't work is because cinnamon is not activating an olfactory receptor, it's actually activating a cold trip receptor instead. And so if you did the same demo but using a cinnamon jelly bean and you held your nose, it still tastes very much like cinnamon because it has nothing to do with olfactory reception. It's actually the taste of cinnamon has to do with these trip channels that are being activated in the skin of your mouth. Kind of cool. Okay, so we've already talked a little bit about pain because we find very high temperatures to be aversive. They're really painful. And so for a long time, at least when I was learning about this as an undergrad, I thought the conventional wisdom was that we were a little confused about pain versus itch, right? So we kind of thought that these were both kind of mechanical senses on the skin, and then itch is kind of just like a low-grade pain. It's now become clear that they are in fact slightly distinct pathways. And so there's a pathway that has to do with pain, right? And so this is mediated by TRPV1 positive neurons that are nociceptive. Nociceptive just means pain sensitive. And so that goes to one circuit pathway. And it goes to a certain type of places in the spinal cord. Itch has its own separate pathway. Now, the mechanotransduction molecules are not distinct. It also uses TRPV1 as the primary sensory transducing molecule. So it's the same one that senses mechanical forces as in pain. However, it has a couple of other properties, and it's actually a distinct cellular population. And so the itch cells are not the same as the pain cells. In particular, it expresses one of these proteins called MRGPRA3. the name doesn't really matter, but I do want you to, I'm just going to put a pin on that protein because that pathway and that family of proteins is going to come back in a little bit in the next lecture. And so I want you to notice that there's other aspects of the circuitry besides the identity of the primary sensory transduction protein that matters in terms of our perception. So in this case, whether or not you perceive something as being painful or itchy has the same primary sensory transduction protein, TRPV1, but because they are activating different pathways, the sensation, the perception of pain versus itch can be distinct. And they have distinct pathways starting from how it gets to the spinal cord and, of course, then how it gets processed in other parts of your body. One more thing I'll point out here that's kind of interesting. About this pathway. So you'll notice here that they are actually talking to each other, right? So there's a pathway here where the pain pathway comes down here, and this neuron is a glutamatergic neuron that's going to release neurotransmitters activating, doesn't matter what the cell is called, this blue cell right here, and this cell is going to actually inactivate the itching pathway. If you've ever had a really itchy patch of skin and felt the urge to kind of slap it to get it to stop itching, this is why it works. It's because the pain pathway, if you can cause a small amount of pain, it actually inactivates the itch sensation in that same patch of skin. So that's kind of cool. All right, so key concepts to understand somatosensation. We've gone all the way from the primary sensors, mostly all the way to the respective spinal segments to which that somatic information goes. And so our somatic senses we learned includes our senses of touch, proprioception, as well as temperature, itch, and pain. And we learned that they project to their corresponding spinal segments in the dorsal roots of the spinal segments, and the ventral roots and the sacral segments innervate the muscles in the same parts of the body, establishing what are called dermatomes, these kind of map of your skin, and different patches of your skin and muscles that go to the same spinal segment. So it has a little architecture like that. That makes sense if you imagine yourself being a quadruped instead of a bipedal animal. These tactile transduction receptors have receptive fields, and we talked about all the different ways that you can actually characterize these receptive fields. These are the features of the physical stimulus, in this case touch, that activate that particular class of neurons. And they can have not just spatial property, but temporal as well as spectral properties as well, depending on what the neuron actually is. Proprioceptive neurons directly measure limb angles and muscle strength, and this is how especially if you close, it's most obvious when you close your eyes. It's happening all the time, even if you don't close your eyes. But most obviously when you close your eyes, it's responsible for how do you know where your body parts are. And this is absolutely vital for coordinated movements of all kinds. If you lost your sense of proprioception, it would be extraordinarily difficult to move around the world with any kind of fluidity and coordination. And mammalian transduction relies on the piezo protein, and the TRP family of proteins underlie temperature and pain sense. This is the subject of the Nobel Prize in Medicine a couple of years ago. And so those are the key concepts for understanding somatosensation. Next up, we're going to keep going on that pathway. What happens after you hit the spinal cord, right? The pain just keeps going up.